In this video we are going to study about nervous system or neural system in human beings. அதுக்கு முன்னாடி நம்ம வந்து மற்ற ஆர்கானிசம்ஸ்ல எப்படி இருக்குது nervous system பார்க்கலாம். First of all, taking Hydra, which is a cnidarian or Coelenterata, the organism that comes under phylum Coelenterata. It has a network of neurons. Okay, coming to the next upgradement, insecta, அதாவது ஆர்த்ரோபோடால எப்படி இருக்குன்னா ஒரு brain, it has a brain with number of ganglia. Ganglia are nothing but group of cell body outside central nervous system. Group of cell body outside என்ன cell body? நியூரானோட cell body, central nervous system. And some neural tissue is present in phylum Arthropoda, that is insects. Coming to the vertebrate, we human beings, we have developed a neural system. Okay va? So basically, histologically speaking, nervous tissue, brain, spinal cord, nerves, எல்லாத்தையுமே எல்லா nervous tissue we can classify histologically. cellular structure வச்சு நம்ம classify பண்ணும்போது nervous tissue has two types of cells. One is neuron and another one is neuroglia. Neurons are highly differentiated cells. Neurons and muscles, both of them are highly differentiated cells. They lack centrioles, which is a cell organelle responsible for cell division. So அதனால என்ன ஆகும்னா என்னதான் டேமேஜ் ஆனாலும் சரி நியூரான்ஸும் skeletal muscles cannot undergo cell division. Okay va? Once they get differentiated, they cannot undergo cell division. Okay va? அடுத்து வந்து neurons can be classified into unipolar, bipolar, multipolar and pseudo unipolar. Okay va? So what is unipolar? Unipolar neuron என்னன்னா இங்க வந்து நம்ம பார்க்கலாம். So this is unipolar neuron. This unipolar neuron is not present in the developed human. Okay va? இது எம்ப்ரியோல தான் இருக்கும். ஸோ இந்த யூனிபோலார் நியூரானில் அதில் ஒரே ஒரு போல் மட்டும்தான் இருக்கும், ஒரு சைடில் மட்டும்தான் அதில் இருந்து ஆக்சான் மட்டும்தான் வந்திருக்கும். ஓகேவா? ஸோ அந்த ஆக்சான் இப்படி வந்து அது ரெண்டாக பிரிஞ்சு ஒன்று டென்ட்ரேட்டாகவும் இன்னொன்று ஆக்சானாகவும் ஆக்ட் ஆகுது. திஸ் இஸ் யூனிபோலார். ஓகேவா? சம் எக்ஸப்ஷனல் கேசஸ் ஆர் தேர். some unipolar neurons are also present in central nervous system. Okay, so this is bipolar neuron. It has a dendrite and it has a axon. The direction of transmission of impulse வந்து from this dendrite to the cell body. So cell body is otherwise called as soma. From cell body to the axon to synaptic vesicle. Okay va? This is the direction of transmission of impulse. This is bipolar neuron. அதுக்கப்புறம் வந்து மல்டிபோலார் நியூரான். இது மல்டிபோலார் நியூரான்ஸ் ஆர் present in central nervous system. This is a typical neuron. It has multiple dendrites, so that is the reason why it is called as multipolar neuron. And there are Nissl granules, right? These Nissl granules are nothing but they are ribosomes of neuron is called as Nissl granules, and it has multiple dendrites and a single axon. The axon contains myelin sheath. Myelin sheath is a insulating thing, so it is formed by a cell called Schwann cells. Schwann cells in case of peripheral nervous system, or otherwise oligodendrocytes inside the brain. Oligodendrocytes. So these Schwann cells, oligodendrocytes are all neuroglial cells. Okay, so they are all neuroglial cells. Okay, they can undergo cell division because they are not highly differentiated cells. Okay, so this is an axon and it has synaptic vesicles. Okay, this is the basic structure of a neuron. Dendrites, cell body, otherwise called as soma. Cell body contains a nucleus, and this is a axon with myelin sheath, and this is axon terminal having synaptic vesicles. Okay, so these are the various types of neurons. Coming to the various types of neuroglial cells. What are neuroglial cells? Neuroglial cells are supportive cells. So they are supportive cells. They are not highly differentiated cells, and they do not transmit any impulse. They will not transmit any impulse. Their function is to support the neurons. Okay, there are 5 types of neuroglial cells. Number 1 is astrocytes. One is astrocytes. These astrocytes are involved in formation of blood-brain barrier because they have this food process. These food process are helpful in the formation of blood-brain barrier. This blood-brain barrier will prevent the entry of some toxic drugs or toxic materials from the blood to the brain. It will prevent the entry of toxic materials and some kind of drugs, large molecular drugs from entering into the central nervous system. So, this is one of the type of neuroglial cells. Coming to the other type, that is Schwann cells or otherwise oligodendrocytes. Both are having same function. Schwann cells also forms myelin sheath. Oligodendrocytes will also form myelin sheath. But the difference is inside the brain the myelin sheath is formed by oligodendrocytes, and outside the brain the myelin sheath is formed by Schwann cells. So, Schwann cells and oligodendrocytes are involved in the formation of myelin sheath. So, the remaining 2 types of cells are, one is microglia. These microglia are nothing but they are otherwise called as macrophages of CNS, central nervous system, because they are having a function of immunity, or otherwise they will engulf the invading bacteria or viruses or any other pathogen. This microglia acts as a macrophage of central nervous system because inside the brain WBCs cannot enter. Okay, WBCs. So, we know that there are 5 types of WBCs. They are eosinophil, basophil, neutrophil, monocytes, lymphocytes. These monocytes will differentiate into macrophage and act as a pathogen engulfing or bacteria engulfing cell. Okay, so these macrophages cannot enter into central nervous system and it cannot provide immunity to central nervous system. So, for the reason microglia are produced, microglial cells are present inside the central nervous system, and it will prevent the brain from any bacteria. Okay, so it will engulf the invading bacteria, virus or any other pathogens. Okay, this is the function of microglia. Coming to the function of ependymal cells. So, these ependymal cells are present in the ventricles of the brain. First of all, we should know what are the ventricles of brain. Brain is not fully a solid substance. It has some empty spaces. We can see this picture. In this picture, this is a coronal section. This is a coronal section of a brain, and it has, we can see there are some space. These are the empty spaces present inside the brain. These empty spaces are lined by a kind of neuroglial cell called as ependymal cells, and these are the choroidal plexus. Here we can see. So, these are the choroidal plexus. These choroidal plexus along with the ependymal cells are involved in the formation of cerebrospinal fluid, that is CSF. CSF is secreted by this choroidal plexus. Okay, this choroidal plexus, we can see the picture of choroidal plexus here clearly. So, this is a actual schematic picture of a choroidal plexus. So, this choroidal plexus contains arteriole, capillaries and a venule lined by ependymal cells. These ependymal cells are nothing but ciliated columnar epithelium. It contains cilia, and this whole thing along with this ependymal cells, this capillaries will secrete a fluid called CSF inside the ventricles. So, this CSF circulates all over the brain, and this CSF is involved in the nutritional supply to all other brain tissues, because brain cannot get nutrients directly from the blood. So, first of all, this choroidal plexus will secrete CSF, and that CSF will circulate all around the brain and the spinal cord, all around the central nervous system. This CSF, cerebrospinal fluid, will circulate, and from this CSF only, all cells will, all the neurons and neuroglial cells will get its nutritional supply. Okay, so this CSF is very important. So, this CSF secretion is one of the important function. So, this ependymal cell is one of the neuroglial cell which is involved in the formation of CSF. Coming to the structure of a neuron. Okay, so a neuron basically it contains dendrites, it contains a cell body, it contains axon and synaptic vesicle. Okay, so this axon is insulated by Schwann cells, and in between in between the insulation, which is otherwise called as myelin sheath, there are some space called node of Runway. Okay, so another one more thing is this neuron does not contain centrioles because it is highly differentiated cells, it cannot undergo cell division and it will not undergo cell division, hence it does not contain centrioles. Okay, centrioles are absent, so no cell division. Neurons cannot undergo cell division. So then how whenever a neuron is damaged, how will it get healed? So for that we can see this picture. Okay, in this picture we can see that the neuron is damaged here. Okay, a neuron whenever damaged, then it can regenerate. That too, the damage should be very small. If there is very small damage, it can degenerate and again it will grow again. So the regeneration will occur. But if an entire neuron or a very long segment of the spinal cord is damaged, it cannot regenerate, and at last what will happen is the neurons that will be present at the damaged site of the spinal cord will die, and there will be no transmission of impulse from the spinal cord to the damaged part. So that leads to the atrophy of muscles and limbs. Anatomical classification. Nervous system can be classified into central nervous system and peripheral nervous system. Central nervous system contains brain and spinal cord. Brain includes cerebrum, cerebellum, midbrain, pons, medulla is brain, and the spinal cord. So brain and spinal cord is central nervous system. So central nervous system can again classified into brain and spinal cord. Brain can be classified into forebrain, midbrain, hindbrain. Forebrain develops into cerebral cortex and diencephalon. Midbrain develops into midbrain only. So hindbrain develops into cerebellum and pons and medulla. Okay, this is the anatomical classification of central nervous system. Peripheral nervous system, if you take, there are this peripheral nervous system includes various nerves that come out of central nervous system. Various nerves that come out of central nervous system. Okay, from brain there are 12 pairs of nerves which will come out. So from brain there are 12 pairs of nerves that is coming out. So they are called as cranial nerves, and from spinal cord gives 31 pairs of nerves, 31 pairs of nerves. So that is why the nerves that comes out of the spinal cord is called as spinal nerves, and it is of 31 pairs. There are 31 pairs. So the peripheral nervous system includes cranial nerve and spinal nerve. Okay, cranial nerves are 12 pairs and spinal nerves are 31 pairs. Okay, so this is the anatomical classification. Coming to the cranial nerves, there are 12 pairs of cranial nerves. We need to study about this cranial nerves clearly because it is having some important functions. So all the cranial nerves come out of the brain. Okay, so first is olfactory. Olfactory nerve is involved in the smell. It is a sensory neuron. This olfactory neuron is a sensory neuron. It is involved in the sensation of smell. So what is meant by sensory neuron? Sensory neuron is a neuron that carries sensory impulse from the receptor to the brain. So whenever a neuron or whenever a nerve carries impulse from receptor to the brain, then it is called as a sensory neuron. Sensory neuron always carries impulse from receptor to the brain. Okay, so this olfactory neuron is a sensory neuron. Coming to the second neuron, that is second nerve, that is optic nerve. First is olfactory. So olfactory is sensory and its function is sense of smell, because it will carry impulse from the nose to the brain, that's all. It will carry sensory impulse from the nose to the brain, that's all. So what is optic nerve? That is second nerve. So it is also sensory. It will take the impulse from the retina. Retina is the layer, photosensitive layer that is present inside the eyeball. So from retina, this optic nerve will carry the impulse to the brain. So here it is also sensory because it carries impulse from the eye to the brain. So it is also a sensory nerve, because here if you see, this is the 10 layers of the retina. Okay, so this is the first layer, and here comes the cones and rods, this triangular dendrite, modified triangular dendrites, right. So this whole thing is a bipolar cone neuron, and this is the bipolar rod neurons. Okay, then coming to the thing, so here these are the photoreceptor cells, these cones and rods. Okay, they will transmit the impulse. These cones and rods are nothing but bipolar neurons, they are present in the retina. Okay, this the photoreceptor layer is the modified dendrite, and this is the cell body, and this is the axon. Okay, so it will transmit impulse, and that impulse will be carried out by the optic nerve to the brain. So it is a sensory neuron. Optic nerve is a sensory neuron. It will carry impulse from the eye to the brain. So it is sensory. Oculomotor nerve, it is motor neuron because it is involved in the movement of eyes. So it is motor neuron. It is not having any sensory fibers. It carries impulse from the brain to the muscles around the eye, that is extraocular muscles. Okay, oculomotor nerve is motor in nature, and its function is movement of eyeball. Coming to the fourth, trochlear nerve, it is also motor, and it is also involved in the movement of eyeball, that is rotation of eyeball. Fifth one is trigeminal nerve. Trigeminal nerve is a mixed nerve because it contains both sensory and motor component. Okay, it is involved in the functioning of facial parts. And then abducent nerve is motor, and it is also involved in the rotation of eyeball. Facial nerve is very important. It is a mixed nerve because it contains both sensory and motor fibers. Okay, it is also involved in the functioning of facial parts. It is involved in the secretion of saliva from salivary glands. It is involved in the muscles of face. It is involved in the stimulation of muscles of the facial expressions. Whenever our face expresses a feeling, right, so some of the muscles of the face are contracting and some of the muscles of the face are relaxing. So for that facial expression, some nerve is involved, and that nerve is facial nerve. And then eighth nerve is auditory or vestibulocochlear nerve. It is sensory in nature because it carries impulse from the ear to the brain. So it carries sensory impulse, that is sound impulse, from the ear to the brain. At the same time, along with that in function, it maintains the equilibrium. It maintains this nerve maintains the equilibrium. Okay, then coming to the ninth nerve, that is glossopharyngeal. It is mixed nerve. It is involved in the taste and touch of the tongue. And vagus is also mixed because it regulates visceral organs like heart, lung, and gastrointestinal tract. Okay, then spinal accessory nerve, it is motor. It is involved in the movement of the muscular movement of pharynx, larynx, neck and shoulder. And finally hypoglossal nerve, it is also a motor. It is involved in the speech and swallowing. It is a motor because it is not having any sensory fibers. It is only simply motor, and it is involved in the speech and swallowing. Okay, so these are the various cranial nerves. Coming to the functional classification of the central nervous system. Functionally, the nervous system is classified into central nervous system and peripheral nervous system. As usual, central nervous system is classified into brain and spinal cord. Brain is classified into forebrain, midbrain, hindbrain. Nothing different here. But coming to the peripheral nervous system here, based on its function, it is classified into afferent and efferent neuron. Afferent neuron is the one that carries impulse from the receptor to the brain. Okay, efferent neuron is the one that carries impulse from the brain to the effective organ or effective muscle. Okay, so from receptor to brain, it is taking some kind of sense from receptor to the brain it is taking. So that is the reason why it is called as afferent neuron. And from brain, it is carrying impulse. The efferent neuron is carrying impulse from the brain to the effector organ or effector muscle. As a result, we will do some function like we will move our hands, or we will have some secretions like lacrimal secretions or salivary secretions. Okay, this efferent neuron again classified into voluntary and involuntary. Voluntary is the one which is under our conscious control. Whenever we are conscious, then only that functions will happen, like swallowing, chewing, or speaking, walking, everything. Muscular movements, generally to speak, all are voluntary in nature. Whenever our brain thinks consciously, then only these functions will occur. But involuntary, if you take, involuntary functions are not under our conscious control. Even though we don't think about digestion or anything, our body will keep on, our gastrointestinal tract will keep on digesting, heart will keep on beating, lungs will keep on respirating. So there is no voluntary control over these visceral organs like heart, lung, kidney, liver, gastrointestinal tract, bladder and all. So there is no voluntary control over these visceral organs. They are involuntary in nature. Okay, they are not under conscious control. We need not think of, we need not think about it 24/7 for it functionings to happen. Okay, this involuntary nervous system can be classified into parasympathetic and sympathetic. Parasympathetic nervous system is rest and relax, rest and relax, because this parasympathetic nervous system is activated whenever we are taking rest. Whenever we are sleeping, then only this parasympathetic system will be active. But sympathetic system, if you take, sympathetic system will be active. Parasympathetic system is activated when we are taking rest, or when we are relaxing. Then only parasympathetic system will be activated, and sympathetic system is activated whenever we are in a hurry. This pictorial depiction of effects of sympathetic and parasympathetic system can be seen here.